So today we will discuss the development of ear and the development of ear. So the first indication for the development of the ear can be found in the embryos, approximately found at around 22nd day, and there is a um, thickening occurs of the surface ectoderm on each side of the rhombencephalon here. That is the myelencephalon. Actually, this is a transverse section through the region of the rhombencephalon, ensuring the formation of the aortic vesicles. This is a picture. Picture is 24th day. That is the 27th day, and that is the 4.5 weeks, almost the fourth weekend of the fourth week. So these techniques and this technique, if you'll we'll see over here, the thickness, the aortic plaque coats are invaginates rapidly and forms the aortic or the auditory vesicle in the 27th day. The aortic vesicle formed completely. There is an invagination of the plicotes. Then the aortic pit formed around 27th day, and around 4.5 weeks, the vesicle is formed. During later development, each vesicles divide into the ventral and the dorsal portion. So the ventral component that give rise to the saccule and the saccular portion and the cochlear duct, while the dorsal portion, this portion, that forms a utricle semicircular canal and the lymphatic duct. So together, these epithelial structure form the membranous labyrinth. So first we'll talk about the saccule, cochlea, and the organ of the cotta. In the 6-week development, the saccule form the tubular outpocketing. Development to form the tubular outpocketing at the lower bulb. Here. This outgrowth, the cochlear duct, penetrates the surrounding mesenchyme in the spiral fashion until the end of the 8th week. When it has been completed 2.5, it will take the 2.5 turns. Its connection with the remaining portion of the saccule is then confined to the narrow pathway, the ductus reunion. Here. So the mesenchyme surrounding the cochlear duct around mesenchyme basically is surrounding the cochlear duct. This is a cochlear duct, and it is surrounded by the mesenchymal cells, and surrounding the cochlear duct. Soon it will be differentiate into the cartilage. In the 10th week of development, this cartilage shell will undergo the vacuolization here, the vacuolization. And the two paralymphatic spaces, the scala vestibuli and the scala tympani formed. So the cochlear duct is then separated from the the cochlear duct is separated. This is separated from the vestibuli by the vestibular membrane, that is also called a Reisner's membrane here, and form the scala tympani by the membrane that is called the basilar membrane. The lateral wall of the cochlear duct remain attached to the surrounding cartilage by the spiral ligament here. whereas its median angle is connected to and partially supported by the long cartilaginous processes, the modulus, the future axis of the bony cochlea. So with the further development, however, they form two ridges, the inner ridge and the future spiral limbus. The future spiral limbus, this one, and the inner ridge. So the future and the future outer ridge as well formed here. So here in this figure, this is the outer cell layer, the inner cell layer, right? And this is the spiral limbus, the inner ridge. The here in this figure, this is your outer ridge. This is your inner ridge, the spiral limbus. The outer ridge form one row of inner and the 3 or 4 rows of the outer hair cells, the sensory cells of the auditory system, and they are covered by the dictorial membrane here. That is a fibrillar gelatinous substance, and it is substances and it is attached to the spiral limbus that rest with its tip on the hair. The sensory cells and the dictorial membrane together constitute the organ of the corti. The impulses are basically received by this organ are transmitted to the spiral ganglion and then to the nervous system by the auditory fiber of the cranial nerve, the vestibulocochlear nerve. If we talk about the utricle and the semicircular canal during the 6th week of the development, the semicircular canals appear. The semicircular canals appear and as a flat out pocketing of the utricle part of the aortic vesicle. The central portion of the wall of this out pocketing is eventually oppose each other and disappear, giving rise to the 3 semicircular canals like this. Whereas one end of each canal dilates to form the crus ampullae. Cells in the ampulla formed from a crest, the crista ampullaris, contain the sensory cells of the maintenance of the equilibrium. So the cells of the cristae and the maculae are as basically as a result of the change in the position of the body are carried to the brain by the vestibular fibers of the 8th cranial nerve. During the formation of the aortic vesicle, a small group of cells break away from its a small group of cells break away from its wall and form the statoacoustic ganglion. Other cells of this ganglion are derived from the neurocrest cells. The ganglion subsequently splits into the cochlea and the vestibular portion, which supply the sensory cells of the organ of the corti and those of the sacule, utricle, and the semicircular canal, basically. Now, if we talk about the middle ear, which is also called the tympanic cavity. And we'll discuss here the tympanic cavity and the auditory tube. So the tympanic cavity is basically originate in the endoderm and is derived from the first pharyngeal pouch. This pouch is basically the first pharyngeal pouch expands in the lateral direction and come in contact with the floor of the first pharyngeal cleft. The distal part of the pouch, the tubotympanic recess, widens and give rise to the primitive tympanic cavity. Here. While the proximal part remains narrow, narrow and forms an auditory tube, the eustachian tube, through which the tympanic cavity communicates with the nasopharynx. So if we talk about the bones of the middle ear, there are three bones basically. What we have discussed in the cross anatomy of the middle ear, that is the malleus, incus, and staves. So basically, malleus and the incus are basically developed from the pharyngeal arches one, that is the Michel's cartilage. And while the stapes जो है वो आपका it basically developed from the pharyngeal arch 2, that is the Richard's cartilage. And the auditory tube and the middle ear cavity both develop from the pharyngeal pouch 1. The tympanic membrane is developed from the pharyngeal membrane 1. This membrane separates the middle ear from the external auditory meatus of the external ear. And it is basically, if we talk about the innervation, it is innervated by the mandibular nerve and the 9 cranial nerve. So external, if we talk about the external ear, so the external ear is basically external ear is basically developed from the dorsal part of the first pharyngeal groove. The ectodermal meatal plug then formed, and later the central cell of the plug degenerate from the cavity, which forms the inner part of the external acoustic meatus. That is the external acoustic meatus. Meatus basically attains full length around the 9 year of age, and the tympanic membrane is derived from the first pharyngeal membrane. Tympanic membrane developed from the three sources: the ectoderm, the endoderm, and the mesenchyme. The ectoderm of the first pharyngeal groove, the endoderm of the tubotympanic recess, derivative of the first pharyngeal pouch, and the mesenchyme of the first and the second pharyngeal arches. So if we talk about the auricle of the ear, so the auricle is developed from the sixth mesenchyme and proliferation at the dorsal end of the first and the second pharyngeal arches surrounding the first pharyngeal clefts. These swellings, okay, basically these swellings are that is called the auricular hillocks. 3 on each side of the external meatus, and later it will fuse and form the definitive auricle. As the fusion of the auricular hillocks is complicated, it's a developmental is a complicated procedure. So the developmental abnormalities of the auricles are common at this time when the auricles are forming. So initially, the external ears are in the lower neck region, but with the development of the mandible, they ascend to the side of the head at the level of the eyes as the mandible develop, and auricle assumes at the normal positions. Thank you.